All right. Hello, everyone. My name is Adhvay Karthikeyan, and my mentor was Ms. Troncoso, who is an undergraduate student at UNC. I live in Winchester, Massachusetts, and my Polygence project is a review paper that outlined the impact of NLRP1 on the pathogenesis of vitiligo. I chose this topic originally because of my father, who was originally from South India, and he had dark skin. As a child, at the age of 5, my father started to lose pigment in his skin and started to lose his identity. After he lost all of his color and he turned completely white, I wondered why this happened and what he could have done to stop it as a kid. Now, as a high schooler, through Polygence, I had the resources to actually research this topic and create a tangible paper from it. As an introduction, autoimmune conditions such as rheumatoid arthritis, psoriasis, and vitiligo pose significant challenges to those affected, with their immune systems mistakenly attacking their own tissue. While the triggers underlying the diseases vary, one common thread among them is a dysregulation of the immune system, often involving inflammasome activation. Specifically, vitiligo is a chronic autoimmune disease characterized by the loss of skin pigmentation due to the destruction of melanocytes. Vitiligo affects approximately 1 to 2% of the entire global population and does not show a preference for any specific age group or gender. While the autoimmune mechanisms have long been implicated in vitiligo pathogenesis, the specific triggers driving the attack on melanocytes have remained unknown. Through this literature review, this paper identified NLRP1 gene as a potential culprit in this autoimmune response. A little bit about the oligomerization of the NLRP gene. Basically, the NLRP1 gene initially codes for a protein that plays a crucial role in the innate immune response. This protein, known as NLRP1 or NALP1, is involved in the assembly of a multi-protein complex called the NLRP1 inflammasome. This complex acts as a scout for intercellular danger such as stress, tissue damage, or like infections. And basically, it gathers adapter proteins such as ASC and facilitates the autoproteolytic cleavage of caspase-1. Activated caspase-1 cleaves these things called cytokines, which such as interleukin-1 beta and interleukin-18, that basically contribute to inflammasome activation. My research basically has shown that the NLRP1 gene in individuals with upregulated NLRP1 gene basically ultimately contract vitiligo because this upregulation can lead to heightened immune surveillance and response deficiency, which ultimately results in the destruction of melanocytes in their body. By understanding the precise mechanisms behind NLRP1 mediated inflammation in vitiligo, we could pave the way for targeted therapeutic interventions aimed at reducing inflammation and preserving melanocytes. The implications of NLRP1 dysregulation extend beyond vitiligo. Like I said before, other autoimmune disorders such as psoriasis and rheumatoid arthritis, the NLRP1 gene has long been implicated in those. Basically, the genetic polymorphisms in the NLRP1 gene have been associated with susceptibility to these diseases as well. And this underscores the importance of further investigating NLRP1 mediated inflammation and its contribution to autoimmune pathologies. In conclusion, by understanding the role of NLRP1 in inflammatory pathways, we should hope to not only advance our understanding of vitiligo, but also pave the way for targeted therapeutic interventions in the management of autoimmune diseases. Thank you.
Okay, great. Thank you so much, Adhvay. We have a pretty specific question here. At the beginning of your presentation, you mentioned that your father has vitiligo, and one of the questions that pushed you to start this project was, what could he have done to stop it? How do you think you'd answer that question now after all the research that you've done and given what you just presented?
Yeah, so actually, when I first got into this Polygence project and we had to first pick our topics, I was kind of set on picking something to do with vitiligo. And originally it was trying to find a cure, but then obviously if it was that easy, someone would have already done it. The best thing that he could have probably done was get out of the sun. A lot of UV radiation basically expresses the NLRP1 gene. So like wearing sunscreen when you're in the sun, I think UV radiation was definitely like the biggest factor that you can control. But other than that, it's just genetics. So there's not much you could have really done.
Okay. Another question we have was, what was either the most interesting or surprising finding that you found along the way?
Yeah, so like I said before, the topic was originally leaning towards how we could have stopped it. But after reading so much literature and going through data sets over and over again, there were mentions in a couple papers about the NLRP1 gene, or the NLRP gene specifically. But the most surprising thing was in all of these data sets, pretty much consistently, the NLRP gene was upexpressed in patients with vitiligo. After they went to the hospital and there was like DNA tests done on them, every single one of them consistently had like an overexpressed NLRP1 gene. So that was really surprising that no one pointed it out as much as like a whole literature review.
Oh, cool. And another question from the audience is, what was the biggest challenge that you faced while performing your research?
Yeah, so vitiligo itself is, although it's a very prevalent disease, like 1 to 2% of the global population has it, it's still relatively unknown. Like I said, the triggers behind it isn't really known that much, and it's not really documented well at all in many countries. They don't really test for it, and normally the doctors kind of just look at their face, and if they start losing pigment, they diagnose it. So a lot of um, a lot of metrics were missing because it's not really well documented in the sense of like hospitals keep track of how many patients they have with vitiligo. So that was definitely the hardest part of my research.
Great. And of course, for me personally, what comes next for you in this research?
Yeah, like I said, I think that we should definitely start documenting vitiligo more specifically, but then like this gene specifically, we should start focusing on it through therapeutics and through other documentation. We should start researching it more.
Great. Well, thank you so much, Advay, for that presentation.